first session here is going to be Dr. John Beever, who's at the University of Tennessee. He's a member of Animal Science and also the College of Vet Med. His seminar will be Biotechnology 101, a practical producer guide to gene editing, vaccinology, and genomics. John, I'll give you this, and I'm going to shut up.
Stay down. We're all candidates, first time. I want to stay down here because it's so the pointer works. I guess you guys on either side are a little difficult because I bet you the pointer. Oh, you can see that. You guys can't see that down there, can you? I think I'm gonna need the pointer, so I'm gonna stay down here so I can see what I'm talking about. Oh, that, I think it's probably in my wheelhouse. But as Mr. Schultz just said, no, everything I'm running up all of a sudden in that, but I may run out, you may run out of answers. So I'm not necessarily the guy that knows everything about it, but I'm gonna try. I think it's important that this is a part of the Young Producers Proposal. These are two topics. So I live in the gene editing and vaccinology. Genomics you're going to hear about throughout the meeting. I think for the past, a dozen or more years at EIF, genomics is now fully emerged. And of course, we do things that you'll hear about this time around, but I don't think there's really need to go into genomics too much. These other two topics are current day topics and things that are being actively investigated from a science standpoint and are of concern to the industry. And you guys are going to be one dealing with it over the next 5 years or 5 to 10 years, and so that's why I think it's long period of time. So let's just start right off. Two topics. Gene editing, the bigger term is genome editing, but usually we're thinking about doing any editing. We're actually looking at a specific gene. I'll talk about that here in a moment. But for today's this session, I want to talk about what is gene editing, how to perform gene editing, how gene editing and artificial reproductive technologies are tied together. And then some examples that are current examples in livestock species, and I say livestock species because I'll use one of them in a pig. So I think a really good example. And then another one that's a little bit futuristic, but I think you probably heard about it in popular press. It's in multiple species. And then I'll spend a little bit of time on the regulation of gene edited animals, because I think that's a topic of interest. And it has a lot to do with whether or not we see the emergence of these types of animals in the industry in the very short term. And what you'll learn is that they are regulated by the federal government, and that regulatory body is the FDA. And I'll talk about some specifics in regard to that at the end of this section. Okay, so what is gene editing? You can find multiple definitions all over the internet, of course, but basically it's the use of modern technologies to precisely change the DNA or the genetic blueprint of an organism. The key word in that definition is modern molecular biology technologies. I'll talk about those, and then precision, precisely change. The concept is not new. We've been interested in editing the genome of different species, different organisms for a long time. And in fact, one of the major components of genomics in general was the editing of mice, laboratory mice, so that we could figure out what lots and lots of different genes did. We go in and manipulate that gene in mice and say, okay, what happened? And based on what happened, we would say, okay, we know this gene does this. Okay, so we've been doing it since 1989. Was one of the first papers published in mice from a genome engineering standpoint. But The fact of the matter is that it was difficult, it was inefficient. Some of the things from a precision standpoint weren't where we wanted them to. You get random integration of the DNA molecules in the genetic material. And it just wasn't something that was really perceived with, actively. So again, the advanced precision versus targeting, previous technologies targeting certain genes. We didn't have it, but now we do. We tend with these tools to have a little bit higher efficiency of successful edits. That's questionable. There's quite a range, but on average, I'd say we do have higher efficiency with these new tools. And these new tools are more amenable to direct editing of germ cells and embryos. Previously, at least to obtain the same precision that we like, we would do these in culture and in cell lines and things like that. I'll talk about that here in a second. But now we can actually do direct editing in germ cells and embryos, and there's an advantage there we'll talk about in a second. Okay, so gene editing technologies. Remember, I guess we use some bigger words, and I don't expect you to remember all these. In fact, these first two, you probably won't remember it all, but the third one, you've probably already heard the popular press a number of times over the last handful of years. These technologies are sometimes referred to as nuclease technology. And the reason why they're referred to that way is that these first technologies, something called zinc finger nucleases and transcription activator-like effector nucleases or catalysts, used an enzyme here called FOP1 to actually cleave the DNA, and that's the definition of a nuclease. It cleaves the DNA. These two technologies were a huge advancement in this precision of editing, in that you could then target based on these things out here at this side and these things here or here and here. You can target a very specific DNA sequence of the genome. Okay, and what I mean there is that you have in a cell, you have about 6 billion DNA base pairs, and you want to go in and precisely modify one or a handful in a single position. Okay. That precision is determined by these. And these are little molecules that recognize particular DNA sequences. When they recognize those sequences, then they bring these enzymes into context where they can cleave DNA. So this is circa 2009. 2008, 2009. This is 2011, 2013, something like that. 2014, 2015. This is called CRISPR. Again, this is one you've probably heard about in the popular press. It's the method of choice now for a number of different reasons, which I'll talk about in a minute. Just FYI, if you're wondering about how people feel from a science standpoint about the impact this technology has. This was awarded the Nobel Prize in Chemistry in 2020, or two scientists were awarded the Nobel Prize in Chemistry in 2020 due to the impact we think that this CRISPR-Cas9 technology is going to have on genome editing. Not only in human medicine, but of course in plant matter species matter. The difference between these two technologies and CRISPR-Cas9 is that this molecule relies on identification of the target DNA sequence by binding something here called the guide RNA. And this is a synthetic RNA molecule. Okay, sure. This is a synthetic RNA molecule that actually identifies the DNA sequence that we're actually targeting. The big key to all these technologies is the idea that at a particular place in the genome, we can make what's called a double-strand break. So we can break the DNA molecule on both strands and separate the two molecules of DNA. Now, why is that important? Here's our CRISPR-Cas9 again. Again, this guide RNA directs the molecule to the position that we want to change. We make a double-stranded break in that DNA molecule. And there are two possible ways to rectify that double-stranded break. Once we have a double-stranded break, the cell actually activates its own internal machinery to say, hey, we gotta fix this. Okay? And so it proceeds to get all that equipment out and begins to figure out how to fix this double-stranded break. Well, part of the beauty of this problem is that sometimes it doesn't fix it exactly right, it fixes it pretty close. But when it fixes it, it might leave out a few DNA base pairs, or it might add a few DNA base pairs. Either of those things, though, will modify the function of that gene. And in many cases, we might be interested in simply disrupting the function of that gene. Okay, and I'll give you an example of that in a little bit here. So the cell goes ahead and repairs that broken DNA, and it can again repair either missing few nucleotides or adding few nucleotides. This is probably the most common occurrence from an editing standpoint. And again, we are oftentimes interested in disrupting the function of the gene to get the desired phenotype we're interested in. The other process, or the other possible resolution is that you will actually use a template molecule, maybe either the other chromosome that's in the cell or a user supplied template. In other words, I can actually put a DNA molecule in with my CRISPR, and I can say, hey, if you're going to fix this, why don't you fix it this way? And I can introduce very specific mutations that way. And so we provide this template, this donor DNA template, and it says, oh, this is what this is supposed to look like, irregardless of repair, just like you want it. Now, what I tell it to change it to may not be what the original sequence was, and of course that's the idea. All right. So how do we actually get this gene editing done, or what do we perform gene editing on? This is what we bring together the gene editing along with system reproductive technology or advanced reproductive technology. We can do it in two main categories here. We can do it in what we call somatic cells. Those are cells that are already differentiated. They can be like skin cells. In fact, we use one of the types of cells that are in your skin called fibroblasts. So I can make a cell line, or I can make some cells that I can grow in culture. So in vitro culture, I can make one of those. I can make a cell line that I can work with in the lab by basically culturing these fibroblasts. This would not be any different than I'm going to mention cloning here in a minute. But this would not be any different than you wanting to clone an animal that you were interested in and sending an ear punch into whoever you chose to make that clone. They would grow a fibroblast cell line from that ear punch, and they would take those cells and produce you a clone animal. Okay. What we're doing is we'll take tissue, whether it be from an adult animal or from say a fetal animal. Those cells grow a lot better, so I would use fetal cells. We can grow them in culture in the lab. And then we can manipulate them with our CRISPR system. And the beauty of this approach is that we can actually pre-select cells that have undergone the modification that we want. So we don't have to, it's not random. We can actually go and screen those cells and say, oh, that cell there has the modification I want. I'm going to use that cell to produce a new live animal. Okay, so that's a big advantage. The disadvantage is to produce a new live animal requires going through the somatic cell nuclear transfer clone. And as of yet, cloning is still probably not as efficient as we would like it to be. It's about 1 in 10 successful pregnancy rate all the way to term. It can be a little bit higher than that, depending on some of the cells that you use, including these embryonic stem cells. But cloning is not quite as efficient still as what we like to be, you know, doing that for over 30 years. But nonetheless, we produce a live animal with our edit via this process. The other approach is to use either gametes, embryos, or embryonic stem cells. The embryos in these cases, particularly in cattle, will be most likely in vitro fertilization or IVF-derived embryos. In that we have to treat these embryos, we have to try to edit these embryos when they're single-cell embryos. And the reason why we have to do that is that once that embryo begins to divide, accumulate cells, we have to make sure our modification gets into all those cells. That tends to be difficult. So if we do it at the single cell stage, then all the cells in that developing embryo will have modification we want. If we do it any later than that, we have the potential of having some cells in the body that have modification and other cells that do not. This is a relatively, I won't say it's a new concept. In fact, embryonic stem cells are what we used in mice all those years. It's been typically difficult to develop embryonic stem cells in livestock species, but there's been some progress made over the last half a dozen years or so. That's now let us take the inner cell mass from embryos and grow them in culture, and these cells remain pluripotent. In other words, we can actually send them through somatic cell nuclear transfer, but because they're stem cells, they actually perform quite a bit better. And using stem cells, somatic cell nuclear transfer, we can get pregnancy we can get term pregnancies up in the 20-25% range. Okay, and so this is not a trivial thing to develop. What's the problem with doing this? Well, the downside here is that we have much less opportunity to select for the specific modifications we want. Okay, and again, I should say here that, you know, as scientists doing this, we're not trying to do it willy-nilly. We're trying to do it precisely so that we know exactly what we're delivering or that we're going to get what we expect. And so because of a number of different things, efficiency, targeting, what have you, we don't have the opportunity to directly select specific edits. We're modifying those embryos, we're putting them in recipients, and we have to see what turns out at the end. Some will have modification, hopefully, some may not, some may have modification that we didn't want to have. Okay. The upside of this, though, is that we can use the most current high merit genetics to do this in we're doing it at the embryonic or the embryo level. So we can take and make embryos from the highest current level, highest genetic merit animals, and we can use that to make these edits in, right? There's no use in going back and using cells from an animal that's 20 years old, right? That would ignore all the genetic progress we've made in 20 years simply to get an edit in that may or may not benefit the industry once we get it out there. Okay. So even though we don't have a lot of control over the edits, we can work with very current germplasm, and so there's the upside there. I mentioned the potential for primary gametes. Okay, so basically we modify either a cell in culture or an embryo or an embryonic stem cell with also in culture. We might select for that change in the DNA molecule and then produce live animal from that. Look. What are we doing here? All right, some examples. What you notice here, so this is genome edited sheep cattle. Okay. Published in 2015. So there's a reference up here. Okay, 2015. So I told you that the reason why we're talking about this subject is that it's on its way and it's going to come to the industry. Well, of course, 2015 is quite a while. And so it's out there, right? So what do these guys do? These guys are interested in in a gene called myostatin. And most of you probably heard of myostatin. If you've not, you've heard of double muscling in cattle. And double muscling in cattle is caused by mutations in this gene called myostatin. Myostatin is responsible for suppressing muscle differentiation. And when you don't suppress that muscle differentiation, you accumulate more muscle cells, which leads to the high muscularity phenotype that double muscle animals have. These guys use talents to then use CRISPR. The idea here is they use these molecules, these highlighted in gray, to recognize this very specific sequence within this gene called myostatin. And you'll notice that all the mutations that are being made, whether it be in the cattle or whether it be in the sheep, are within this little red box here. Okay? So we're not making any mutations anywhere else. So I told you there were 6 billion of these little letters in each cell of the body. We're limiting where we're making changes to these 10 or 12 nucleotides in the DNA sequence, right? In this case, they weren't worried about making a specific change, they just wanted to disrupt the gene function. So they wanted to knock out, what we call knockout function of this myostatin, so it would increase muscularity. And so if you look here, they removed 3 nucleotides here, removed 3 here. This was a little bit bigger change. This is down here in sheep. They removed 3 here. Each of those wind up having an impact on myostatin function. That being that it disrupts this function. What we got? So these are the 2 Lighthorn Lori animals that were generated from this project. The heifer, which is this one here, did not get edited, and the bull got edited. This is the heifer here on the left-hand side, the bull on the right-hand side, and yes, I know that you expect more muscularity in the bull than you do a heifer, but I think that you can agree that this bull has got a tremendous number of amount of muscularity compared to his heifer temporary. Okay. And so this is a successful edit of this myostatin. This animal has loss of myostatin function and increased muscularity. And then he's propped up a little bit here, but you can see this hindquarter on this little lamb. And of course, he's got a little round pattern hindquarter there, which means that he also has this increased muscularity. Okay, so we can do this. Okay. No, this is 2016. I actually had the privilege of being involved in this research. This is an edit that actually makes cells within cattle resistant to the leukotoxin that's produced by the organism mankind hemolytic. And mankind is the #1 bacterial problem in bovine respiratory complex. Okay. It's an opportunistic bacteria and gets deep enough in the respiratory tract and secretes leukotoxin. They call all kinds of severe pathologies. And it's supposed to say that mankind may have cost the U.S. beef industry probably $600 million a year. Okay, so it turns out if you substitute 2 nucleotides in these cattle, you can make them completely resistant to leukotoxin pathogenesis. In other words, the leukotoxin secreted by mankind doesn't kill the leukocytes that infiltrate the lung tissue, and therefore the animal has a chance to recover. And this is a corresponding paper that tells us that if you change those 2 nucleotides here, you can get to resistance. You can actually find naturally occurring mutations that cause both resistance and higher susceptibility. You go and look at it. Okay, and so we can again gain you information here too. This one I want to talk about because I think it's particularly down in the southeast. This is a paper that, again, this was published quite a while ago. 2014. This is actually a natural mutation. It's called split mutation. It's naturally found in simple cattle. Simple, of course, are tropical breed cattle. But it was discovered at some point that these cattle had the ability to slip off their hair growths real quick, or real easily. So temperature would rise, they would slip down their hair, and they would be more thermal tolerant. So this has been known for 30 some years. And this was a project done at Florida, where they took simple semen and they bred it to Holstein cows, and then graded those Holstein cows back up to what would be a modern Holstein. Okay, think about how long that would take. All right, so I'm crossing simple with the Holstein, and then I'm going to breed that Holstein back up to where she produced milk just like Holstein. How many generations do you think that takes? No guesser? I bet everybody's super right. 5, about 10. Okay. The show was going on 15, 16. We cross back. Okay, 15, 16. Question is, can you ever get them pure, right? Can you ever make it pure Holstein? Well, when they did that, here was the outcome. This is rectal temperature of cows that have the mutation down here, and cows that don't have the mutation throughout the day. These cows stay at a degree and a half cooler all throughout the day. Okay? Mutation works. A few more data points. Respiratory rate. They have lower respiratory rates. Up here, lower respiratory rates. Lower temperature, you saw that. Higher sweating capacity. Okay, all good things. Production. This is the normal cow during the winter time. Lactation. This is a split cow during the summertime, or this is the normal cow during summertime. The normal during winter, normal during summer. Summer slump of lactation. You think we get that in our beef cows? Sure. These are split animals down here. No difference between winter and summer. Okay, so they maintain that lactation potential. The reason why the line is not so clear is there were a lot more cows in this graph than in this graph. But there's no difference from a heat stress standpoint. They still maintain their lactation potential. Okay, so now this data says I would like to have simplification in my cows, but I don't want to go through the generations and generations of breeding in order to get my animals back close to purebred. And in fact, according to some registered germplasm, you can never register those stock after doing something like this. But with gene editing, I can move that mutation into any breed I want to move it in. And that was done for the first time back in early 2022. It was done by a company called Vaccinology. They did it in South America. They've now replicated that in a couple other countries, in the United States. That mutation has now been placed in at least 2 breeds, if not 3. And it's not out there commercially yet, but it probably soon will be. But the idea here is that we don't have to go through that process of breeding. We can make that mutation. It's the same mutation that was made that occurred naturally in simple cattle, and these cattle slip off just like simple cattle. The reason why I put this up here is I mentioned here in about 2 slides these animals are regulated. They're regulated in the United States by the Food and Drug Administration. I'll tell you why that is. But this was a news release where the Food and Drug Administration decided that they were going to use something called enforcement restriction and not regulate these animals. Okay, so that one is a commercial viable animal coming to you sometime soon. Okay, a couple more goodies here. This is probably the one that's got the most potential. It's been licensed by a commercial entity. But this is in pigs. For those of you that don't know, the pig industry is plagued by a virus called 400 respiratory syndrome virus, the PRRS virus. Again, it's roughly $1 billion or more per year in the United States cost. Probably $2.5 billion if you include Europe and the United States. And so we really like to fix that problem. Second half of our talk today again is about vaccinology. They'll say, well, why didn't I learn how to vaccinate for PRRS virus? Well, it turns out that that's one of the pathogens we've not been very successful developing a vaccine for. And so this is a permanent mechanism to alleviate issues with that virus. These are just some data, but this to show you that when you infect these animals, they don't develop fever. This is also a percent of pigs with fever. You see there's none of them over here. PRRS virus kills pigs. So you can look at infection here. The animals that were gene edited, no problem, they stay 100% viable. And then by the time day 14 rolls around, the non-edited ones, they're all dead. Okay? So again, it works. And then there's a couple of data points on these animals don't replicate virus, and you can't find the virus in their circulation. And the reason for that is that when you make this edit, the virus cannot attach to the cell into the cell. So the virus winds up getting clear almost immediately on infection and doesn't replicate in the host animal. Again, it's commercially licensed by genus, and they're in the process of getting FDA regulatory approval for before we wind up seeing the commercial production. And here's the last one. I won't spend very much time on it, but this is one that's been at Washington State University in multiple species, including cattle. This is the idea where you can knock out this gene or you can disrupt this gene nanosubstitution. Nanosubstitution plays an essential role in the development of testes or sperm stem cells. And so when you knock out this gene, what happens is that there's no sperm cells grow in these animals. They have completely normal testes. The upside of this is that you can take sperm stem cells out of a different animal, put those into the testes of these animals, and within about 3 months, they'll begin producing sperm from that donor cell. Right? So if I want to make multiple copies of my best bull, I can take sperm stem cells and I can inject several atoms like this with his sperm stem cell, and they will all produce sperm just like he was producing themselves. Okay. This one's also going through federal, going through the approval process. But probably coming to you real soon, probably in 5-10 years at least, if not earlier. All right, so I talked about regulation. The FDA is a regulatory body. That authority is given to FDA by the Federal Food, Drug, and Cosmetic Act. And basically that says that anything you do that changes the form or function of a living organism, animal or human, they have purview. Okay. It's been a tough go with FDA in that they've not known how to handle these technologies. And it's been the approval process has been relatively ambiguous for a couple decades now. The good news is that they're finding, and you'll see in the next couple of slides, that we are right on top from the topic standpoint. This is all information that was generated in May of this year. This is a definition of what we're talking about. FDA calls these intentional genomic alterations, IGAs. And that terminology is to kind of get away from the technology, the transgenic GMO technology. These are different. These are different types of animals. We're making intentional alterations in the genetic material of these animals, so we call these guys IGAs. And IGAs define how the FDA defines them. The regulation is defined by two things called the guidance for industry, 187A and 187B. These define how or what will be regulated by the FDA. Notice here, this was released in May of 2024. This is 187A. This defines how animals will be evaluated for commercial use by the FDA, and they call it a risk-based approach. And the reason why they use what's called enforcement discretion on slick animals, for instance, is because we already know that mutation exists in a population of animals. Moving it to another population probably has little to no risk. Okay, and so they were deemed to have no risk, and that's why slick cattle are now available. Okay, again, note that this was published in May of 2024. The first publication was came in 2009. It was updated in 2017 with the advance of these new technologies, but it's still been a real hard to pin FDA down as far as what they want. Here's where the crowd can participate. You don't have to participate now. This is 187B. This actually gives you a definition of what the approval process looks like. And again, published in May of 2024, but you'll notice here it says draft in the little red box here. The reason why it's a draft is that this is now published with public comment. You guys are the public. If you need good reading material, it's about 30 pages. That'll put you to sleep after a page and a half. So you got plenty of time between now and July 31st to get through it. But what you have the opportunity to do is to educate yourself about gene editing and livestock and tell FDA how you want that to go. Okay. I think this last slide of the section, I probably won't go on time anyway. This is a matter of national competitiveness. This process regulated by the FDA is unique to the United States, not only because we only have an FDA, but because other countries are just not regulating some of these technologies. And that leaves them, we feel, with a competitive advantage. Okay. And so we want to know what the FDA wants to do from an approval standpoint, and we want to know what data they need for approval. And you are welcome to provide your comments as producers to what you think about these technologies. And I encourage you to do so. Here's the website down here. I know you won't have copies of these, but if you look at CBM GFI 187B on Google, you could probably get here. But do take your time and read that. Okay. Last important thing. So this is also released in May. You'll notice, well, maybe you don't know this. Again, regulation of animals is FDA. Plant microbes is actually USDA. And you think, well, why? What's the difference? Well, again, it's the Food Drug Development Act that defines that review. But now we actually have a memorandum of understanding between USDA and FDA, where there'll be transparency and communication, and perhaps the ability to push some of this through a little bit, as far as risk assessment, things like that. Of course, we also have to decide what to do with it. USDA is in charge of Federal Meat Inspection Act, which oversees FSIF, food safety. And so they're of course interested in how to treat these animals as they might in the future. Okay. All right. Oh, yeah. We're way. Okay, vaccinology. Current topic of vaccine development. Basically, mRNA vaccines. You've heard about them for the last 3 or 4 years. They were the vaccines that helped us with the COVID pandemic, or at least 2 of the companies generated these type of vaccines, and were the most widely used vaccine. Just briefly, why are we vaccinated? I think I don't have to spend too much time on that. Types of vaccines, you never thought you'd come here to gather some people learn about the simple law of molecular biology. I'll share that with you. And then just tell you what an mRNA vaccine is, just, top it off. Okay, we'll go over this real quick. Again, current, I'll just use this Google search. I Google search livestock mRNA vaccines, and there's thousands and thousands and thousands of links that come back. You'll notice that, there's quite a presence here. I encourage you guys to read these, the UT Beever by Dr. Strickland, here on campus. Tennessee Farm Bureau, these are publications to actually, kind of slow down some things that were being said in the public that were absolutely false. And in fact, there's still legislation on the books, in Tennessee and a few other states, about mRNA vaccines and livestock species. And the truth of the matter is, I shouldn't say that because the second link there is actually using mRNA vaccines for influenza in dairy cattle. So you know, for the last 3 or 4 months, that they've popular press has shown the influenza infection in dairy cows. They're worried about the zoonotic transfer of that to human workers. And so people are using an mRNA vaccine in dairy cattle to maybe stave off the influenza problems. The idea here is about half of these issues are something like this. This is from Australia. Maybe I can't blame them because we're on the other side of the world. Title of the article, mRNA-free meat. And this article goes on and on and on about how all the meat that this company provides is mRNA-free. And that is impossible, but that's the position they take. And what that means is that they've not been treated with an mRNA vaccine. But we kind of need to put, I don't know. Some people don't pay attention to the facts, but anyway, we'll try to straighten that out here in the next spot. But that's why we're doing this. Why do we vaccinate? Well, that's why we vaccinate. We have lots of organisms out here that threaten the health of our cattle, and when our cattle come in contact with either viruses or bacteria, there's a good opportunity that they might end up like that. Okay. Alternatively, what we can do is we can use our vaccines. We can administer them according to DQA standards. Okay. And the idea here is that we're stimulating an immune response that, in this particular case, results in the production of antibodies to give a immunity for animals. So once they come in contact with these organisms, these antibodies clear that organism from your system. And they can survive and in fact, not even get sick, is what we hope. This little graphic here, most of you are familiar with giving vaccines. You oftentimes give a vaccine, you give a booster vaccine a couple weeks later, 2-3 weeks later, or a month later. And that's to this primes the immune system. And then this actually amplifies that immune response and generates the secreted antibodies for protection. Okay. Lots of different types of vaccines. Just go over a couple of them here. You guys can use modified live. Most of those vaccines are viral. Basically, what that means is it's a live organism, but it's either been chemically treated to be weaker, or it's a non-pathogenic version of that organism that looks just like the pathogenic version. These are the upside of these, and because they're live, if you infect our cells, they replicate, they hang around a little bit longer, we get a little bit better response. Many of you looked at your vaccine labels probably. Modified lives often only require 1 dose because they hang around a little longer. Killed, on the other hand, oftentimes they require 2. Our immune system does a pretty good job of handling those things. We give the injection, it responds, clears it out. We give another booster, it responds, clears it out, but then we're left with that memory of immune response. This is a little bit of a bit here. There is an mRNA vaccine that is, approved for livestock, from American Animal Health. It's an on-demand prescription vaccine for several different viruses. One of the upsides of mRNA vaccines is that you can evolve them very rapidly. And I think you probably heard about that with COVID. Every couple months, there was a new version of COVID that came out. You were told that there was new boosters being made. I don't know if you know it, but our flu vaccine, like right now, flu vaccine being manufactured now is last year's version. The viruses that we detected last year. If there's a new version of that comes this fall, too bad. Our vaccine doesn't protect us against that. Okay. On the mRNA, on the other hand, we know the sequence of the pathogen that we're interested in, we can modify that vaccine and generate a new vaccine very quickly. Okay. So that's why on a prescription basis on you're a producer, you have a problem with a couple different viruses and big production system. America go in and make the vaccines specifically for those viruses. But anyway, and I guess the other thing I missed that vaccine is not new. Up here at the top, you notice the first smallpox vaccine was generated in 1798. So we've been doing it for a while. Okay. I think it's just a natural evolution of vaccinology. All right, so not going to go through this a lot. We vaccinate, which means that basically we're putting a foreign material or an antigen into the animal, giving it something to respond to. Okay, what our immune system is good for is detecting the difference between self and non-self. Okay, we put that protein or that some of them are glycoproteins. Other things, the majority are proteins. We put that protein in, the system doesn't recognize it itself, and it does these things in order to combat that. And basically, the antigen or the protein is taken up by these things called antigen-presenting cells. They interact with these things called T cells, which help activate B cells. This is a very simplistic, one of the simplistic paths. But eventually you see out here these things called plasma cells that are the ones that generate the antibodies. Okay. Well, the point of the slide here is that we need to expose our immune system or our cat's immune system to these antigens in order to develop this immune response. Okay, so now I'm going to play out the mRNA vaccine. First, we'll go through the central dogma of molecular biology. This was post the discovery of the molecule structure of DNA. But the idea here is that DNA is the molecule that contains all the genetic information. In order to express the information in that DNA, we have to first turn it into something called RNA. Okay, so we have machinery in our cells that reads the DNA and makes an RNA molecule through a process called transcription. Okay. Then that RNA is shuttled out into the cell, and when it gets in the cell, there's these things called ribosomes that take that RNA molecule and translate it into a protein, or they decode it into a protein. Okay? These molecules that the ribosome translates are called messenger RNAs, mRNAs. They carry the message of that information out into the cytoplasm where it can be translated into a protein by the ribosome. Now, let's go back a slide. Remember, most of these things are protein, so we want to expose our animals to a particular protein. So the idea here is we generate this RNA molecule, let our cells turn it into a protein, and then have them respond to it. And in fact, that's exactly what happens. Okay? I've already been 10 minutes over and Dr. telling me 5 minutes. We'll get there. mRNA vaccine is pretty simple. We can make RNA synthetically. So we can do this on an instrument, we can do it on a machine. All we gotta know is the DNA sequence of the molecule we want to make. In COVID, we made mRNA vaccines against something called spike protein, which is the major protein that's on the surface of the virus. That's what our immune system will respond to. So if we know the sequence of that gene, we can make this mRNA synthetically. It's going to have certain properties. It's going to have this thing out here, this thing here, and it's going to have the sequence in here that encodes protein. You make these with some special ribonucleotides that increases their stability. That when you put them in the cell, they don't degrade right away. They hang around a little bit. But the idea here is that we put those in the cell. In order to get them in the cell, we have to encapsulate them in a little lipid membrane. And that lipid membrane is very similar to the cell membrane that we have for all other cells. When you put that, when you inject that in, it merges with the cell in your body, releases the mRNA. That mRNA is translated just like we expect it to be. And then those proteins are actually displayed on the surface of those cells for our immune system to actually respond to it. Okay? So this is just instead of putting in the protein like we would with a normal vaccine, or putting in the entire pathogen, modified pathogen like we would with a normal vaccine, we're actually kind of cutting to the chase. We're putting in this RNA molecule, letting our cells use that RNA molecule to produce the protein of interest, and then they can display it, and our cells can respond to it. Okay? Lots of advantages in mRNA vaccines, especially in regard to responsiveness, changes or evolution in a pathogen. Like I said, we can change those really quickly. Once scaled, they're easy to produce. They're more expensive currently, but as technology changes, they'll probably be cheaper. But the idea here is we have to understand what our what we can expose our immune system to, so that we can get a result where the animal is protected. We have to do that for both of these things. Right now, traditional vaccines are pretty cost effective to produce. But again, once this technology gets to run, there's a lot of upside to mRNA vaccines that I think that the livestock industry might be able to benefit from. I think that's the last slide. We don't have time for questions. I did that on purpose.